The panel period now. Again, we're going to have a two-part panel. The first part is going to really focus on why do we care? How does this stuff really relate, and implications to research, design of research, interpretation of data from research, obligation, responsibilities, and then hopefully a little bit of discussion around resources. The second part, which will be moderated by Ellen Silbergeld, will focus on, unless Ellen doesn't want to, because you're also summarizing, so we can discuss that, we'll focus on the policy implications. Okay? So for the first panel, we have Mildred, as well as, Vince Cogliano from US EPA, Lisa Chadwick from NIHS, Akbar Khan from DOD, and Dan Sharp from NIOSH, sorry. And then Jeanette as well, Jeanette Thurston as well from USDA, sorry. All you agencies. So I think we stay put like we did yesterday, and I'm going to start with Jeanette and sort of move around the table. Are you okay with that? Oh, sure. Okay.
So can you hear me? I'm Jeanette Thurston. I'm with the USDA, but the agency I'm with is the National Institute of Food and Agriculture. We recently organized, we used to be CSRES, and probably no one in this room even knows what that stands for. I don't even know if I know what that stands for. You would know, wouldn't you? All right. So thank you so much for having me join you. This has been a very interesting last couple days. I actually come at it from a very different angle. I'm at the National Institute of Food and Agriculture. We're the extramural research branch of the USDA. We actually, for the first time in 2010, actually funded some microbiome type research looking at how the microbiome of the gut of cattle influences the colonization and shedding of Shiga toxigenic E. coli. And actually, one of the authors that has been highlighted in your references, Dr. Andy Benson, actually received that award, and he'll be expanding that mouse research into cattle. So we're very excited to be funding Dr. Benson, and then we're very interested to seeing what he finds in cattle. So, looking at these questions, I'm not sure I'm the person to answer all of these, but when thinking about the microbiome and thinking about how we view ourselves as humans, this brings a whole new definition of what human means, right? And I think the general public don't understand, and I think a lot of us have already discussed earlier yesterday about microorganisms and how the general public thinks about microorganisms. In my previous job as a researcher with ARS, I would go out and work with producers and do sampling on their farms, and when they would ask me what I'm doing, I was very forthcoming and told them I was sort of sampling for pathogens in the manure. Yes, I am a manure pathologist. Anyway, but what I would commonly get back is, oh, bugs like flies? They don't understand. And if they do understand when you say microorganism, the first thing they think are germs or pathogens. They think very negatively. So when I think about public perception and how important public perception is to driving the science and driving the funding that we have in our agencies, we need to think about, I always think about mom and dad. I always think about how do I relay what I do to my parents and how to describe that. And not only on CNN or other Fox News, God forbid. No, anyway, sensationalism seems to sell, unfortunately. But another thing I was thinking about while I was brushing my teeth this morning, because I knew I was going to have to answer these questions, I was thinking about my daughter. I have a 20-year-old daughter. Back when she was young, EPA, I believe, had a very big I think it was EPA, had a very big education program teaching kids in schools to turn off the water while they brush their teeth. And let me tell you, that really worked. So when I think about public perception and educating the public, not only do we need to think about what's on the news or what kind of commercials we have, but we also have to reach out to our children and make sure it's part of the education of our children as they grow up. So that's the answer to the first question, or at least that's some of my ideas for the first question that we have there. And how we can incorporate aspects of the microbiome research into epidemiologic or occupational studies. At least in our agency, we have been able to start looking at microbiome research, maybe not in humans. I mentioned the work that we had in cattle. We also have a program, it's the second year going, and looking at the microbiome of plants and trying to understand how foodborne pathogens are affected by the microbiome of the plants, either through attachment, persistence, or internalization. So certainly microbiome can be put into other programs. For example, these are food safety programs. I don't know if I'm answering that one very well, and that's what you had in mind. And then in thinking about question 3, I'm going to skip question 3 because that's certainly not my area. On question 4, now this is a very interesting and I think a very complicated question. Certainly, how do we see in Dr. Benson's paper that genetics has an important or significant role in the microbiome. As far as cultural and geographic differences, do we really know? Has there been a lot of information to look at differences around the world or cultural differences? Of course, cultural differences will you have different diets depending on different cultures and different ways of living. So certainly that would have an effect too, I would guess, but I don't know that the research is out there. Again, this is not exactly an area that I've been working in. So that's all the comments I have. Thank you.
Obligated to answer all the questions, but the idea was to motivate discussion. So I'm actually going to jump from here to that end of the room and go to Vince Cogliano next from EPA. Is that okay?
Okay, thank you very much. First, I'd like to congratulate the Academy and the committee for sponsoring this discussion on this topic. I think it's going to do a lot to start to raise the profile of this area of research in the minds of other scientists, regulators, and the general public. And so it does go to the first question, public perception of risks. I don't think this is on the public's radar screen. So I don't think public perception is really affecting any of this, but I think it should go the other way around. I think that it's going to be important that the public understand that this is important because as budgets decline and there's pressure for research money, this could be an area that would be easy to cut out if nobody thinks it's an important area of research in the Congress or other places that are making funding decisions. Now, as to incorporating aspects of microbiome research, I think that we've already heard a few things here about taking more biological samples from epidemiological studies in the National Children's Study, for example. That would be very important. I think there's some other things we can do too. For example, the difference between the microbiome of a child delivered vaginally versus one delivered by cesarean section. We could now just in any case control study ask, what was the circumstance of your birth? Were you and that would tell us, I think, something about the microbiome, and that would be very simple to just add a few, and there's probably other examples that we didn't come up in this meeting, but would suggest questions to put into case control studies or something to put into a questionnaire in a cohort study. Let's see. I would like to talk about number 3, the absence of microbiome information in interpretation of toxicity testing results. Regulatory agencies were putting a lot of effort into understanding the sequence of mechanistic events that leads to disease. And it looks like we've been ignoring a big number of these events that could be there. So it's in some ways like trying to write a novel without using any prepositions. You can put some coherent thoughts together, but you can't tell a story that's very complex or very interesting. And I was also thinking about something I heard Linda Birnbaum say at a previous workshop a few weeks ago, that one person's negative results do not negate another person's positive results. And maybe when we see what look like conflicting results, there are some unexplained variables that if we really knew something about them, that might put into context why some studies are positive and other seemingly equivalent studies are negative. And this looks like a whole set of variables we've been ignoring and may explain some of the conflicting data interpretations we have. In doing that, though, it's going to be important to keep this rather simple. I think that if we try to talk about the tens of thousands of different microbiome measurements you can get, you're going to have a hard time really interpreting them. I think some summary measures, like the differences between the phyla of bacteria with obese people and non-obese people, some kinds of summary measures that we really can associate with disease conditions. What Mildred said, what's your disease? I think we do need to try to initially look for summary measures to be able to try to explain them. And the fourth one about differences on testing abroad or even testing in one part, I was really impressed with the level of differences, the extent of individual variability that there are here. And it means that there really could be very strong differences in the metabolism or the processing of chemicals like arsenic in other countries where the microflora might be different. And so I think if I were FDA, I think they would be very wise to, in any drug that's tested primarily abroad, to do some very vigorous monitoring when it's marketed in this country of the efficacy and the side effects to see that it really does correspond with what was observed abroad, because we could be getting some different results here. And I think it almost it would be wise to do this anytime some new drug is used in the general population, to require some kind of follow-up on each person who takes that to monitor efficacy and side effects. So those are my comments.
Thank you. We're going to come back at the end and sort of really have a sort of a realistic discussion about what do we do in terms of designing the future batch of studies, or even studies that are just starting, like the National Children's Study. But Lisa, you go ahead next, and then we'll go to Akbar. Lisa first, and then you.
Hi, I'm Lisa Chadwick. I'm a program administrator in the Division of Extramural Research at NIEHS. And I just wanted to sort of echo the excitement that Ellen expressed yesterday at the end of the last session. I think the talks that we heard yesterday, particularly in the last session, really illustrated the importance of the microbiome in environmental health research. I mean, this is really our first point of contact with the environment. And we also heard from Tom's talk about how the microbiome plays such a critical role in the processing of environmental contaminants. So one thing we're really interested in at NIEHS is understanding how individuals respond differently to exposures. What makes one person more susceptible to adverse health outcomes from an exposure than another? And, you know, genetics has really not been giving us all the answers, and that really pains me to say I'm a geneticist. But I think we have to really start considering encouraging people to consider the role of the human microbiome in this. And how does the microbiome interact with environmental chemicals? How do exposures, especially early life exposures, which is something that we're particularly interested in, affect the composition and then the later function of the microbiome? This is really a vastly understudied area. You would think, given from what we've heard today, that we would have a lot of research here, but people have alluded to how we had a planning meeting a couple days ago for next steps in the Human Microbiome Project, and Lita showed a graph where she broke down research investments in microbiome research at the different NIH institutes. And NIEHS, we have one grant in this area. So I think our challenge now is to try to encourage people, our investigators, to start thinking about this, this problem. That's really what I have to say.
I am from an agency called Defense Threat Reduction Agency. It's part of DoD. The section I'm in, agency's mission is huge. It's a combat support agency, but I work as a science advisor and program director. And the area we are in is we have to protect and defend against chemical and biological threat agents. So chemical agents meaning that all the chemical agents Army and mostly Army has, and then biological agents are infectious disease agents out there. So this area is very interesting for us. We don't have programs yet, but let me explain it to you why. Because when troops get deployed, especially in different parts of the world, we like to know before their deployment what their microflora looks like and after. And especially in the vaccination programs and stuff, we have noticed from time to time that different troops behave, different soldiers, you give them vaccination, they behave differently. And it has to, microflora has to play, microbiome has to play a role in there. This very, after seeing all the wonderful talks yesterday, it is very apparent that it's going to play a role in there. So if we can take and analyze those things, then it should be very interesting to dissect out all these effects out there and understand in advance. Also, we develop a variety of countermeasures, vaccines, and drugs to protect against these agents. And it should be, microbiome is going to play a big role in there to dissect out before giving these countermeasures to protect against these agents. And understanding will help us to dissect out and protect and defend more better in future. So that's my comment. Also, I mean, I think that at this point, the public perception is this area is such a young area, and it's going to be expanding more and more. And I think public perception is not there yet. I think in the hospitals and stuff, if you ask doctors, I think they know, but outside world is not as aware yet. So as we move along, I think this area will be a huge impact on the policies and also at the public perception in a positive way, I think. So these are my comments.
Thank you, Akbar. And Lita, before I hand it to you, I want you to maybe contemplate and talk a little bit about, I mean, we heard yesterday and today about the impossibility to really separate the interactions between environmental exposures, the microbiome interactions, and the exposome. Steve talked about yesterday. So if we are going to take your meeting and this meeting seriously and build on this, and these things happen so slowly, so even if we have that discussion today, change is going to take forever. But what are some constructive movements we can start to take to bring the agencies together, and at least for the next batch of non-existent money that comes in, think about how we're going to do this differently? So I want to get to the really hard questions. I think there's tremendous kumbaya in the room, but I want to move it into what do we really do beyond acknowledging that this is an issue.
So actually, could I address the public perception?
Yes.
Because I actually have some positive experiences. So as my job as coordinator, I often get asked to give talks to all kinds of groups, not just scientific groups. And I have actually been quite pleasantly surprised that people get the microbiome. And what I mean by that is, I mean, they may not have a deep understanding of it, but for reasons that are yet not clear to me, when I give public talks, people get it. They get that microbes play some role in their lives. And they're very curious about it. They also get creeped out by it. But the fact is they get it. And I don't know if it's because of the probiotics in yogurt or whether because they've been using all these antimicrobials, so they've kind of gotten the idea that maybe they're going the wrong way. I don't really know. I haven't been able to put my finger on it. And the most recent talk I gave was to, there's an annual summit that's sponsored by the Black Congressional Caucus, and it's a health disparity summit. And so in that room, 100 people or so were, policymakers and physicians and all kinds of people. So I asked the convener, well, what level should I aim at? And they said, well, just give your good, broad, introductory talk. And everybody got it. And I got requests to come to their churches and other places to give the same kind of talk so that their family and friends could learn more about the microbiome. So I'm just telling you that I think there's something out there where people get it. How to apply it is a whole different story.
And let me just add to that I was visiting the MIT Museum, and I got my son a huge stuffed animals, the most cutest thing I ever saw. And I thought it was so nerdy, and then I came out and saw KB Toys had it too.
Oh yeah.
So it was everywhere.
It's Giant Microbes, I think is the name of the company that sells these. Great. And unfortunately, I mean, I've actually been giving them a lot of grief, this company, because they're all pathogens except for the Martian microbe. There's no beneficial microbes in that collection of stuffed microbes. And so I've actually been one person kind of campaign to get them to include some beneficial microbes. Okay, let me set that aside. So again, I think in my very fortunate position, I've been asked to serve on a number of working groups and gone to a number of workshops. So I want to point out, so far I've gone to an O&R workshop where O&R wants to develop some kind of way for their war fighters to be able to survive in some kind of dangerous environment if they're thrown behind enemy lines or whatever. And I think they had all kinds of ways to try to augment their war fighters' diets or take a probiotic pill or something, some way so they could actually, I think in one case they were toying with the idea of having them ingest a capsule of nitrogen-fixing bugs that could digest lignocellulose, they could eat wood. There was all kinds of ideas being thrown out there. Coming up in 2 weeks, though, Walter Reed Hospital, the research arm there, they actually have a wound microbiome workshop coming up because, and I think the thing that really pinged them was in Afghanistan when people were getting injured, they were being airlifted out and taken to Germany, and when they were culturing out microbes, some of these were Afghanistan desert microbes that had nothing to do with the human body. And so they're concerned, there are a lot of concerns, not only one about the internal microbiome, but actually the environmental metagenome, what are they getting exposed to in the battlefields, and how does that affect their capacity to recover from wounds? So there's a lot of different interests. A third example I want to provide is, and I mentioned to, I think it's Jeanette at the end, right? Okay. Way above us, the directors of USDA, NSF, and NIH have decided that they want to develop some joint activities, okay, around 4 thematic areas. And frankly, 3 of the 4, even 4 of the 4, in my mind, don't really separate. Nutrition, obesity, plant genomics, and microbiome. Now, I don't know how you're going to get the first, second, and fourth away from each other, but basically those are the areas they want to cover. And I think they've been in conversations for a long time, but we've, I think, finally had our first formal all-day meeting to try to talk about how are we going to go about developing these ideas? Is it going to be new money, or is it going to be already existing programs that we're going to just put new names on and call them joint? And there's also the actual kind of mechanics of doing joint activities, because the review process is very different across those 3 agencies. Nonetheless, we certainly have lots of excellent examples, and I can give 3 already, where there have been joint activities between agencies. They all happen to be microbial. The Microbial Genome Sequencing Program has been a long-standing, 10-year-long until 2 years ago, program between USDA and NSF. Ecology of Infectious Diseases is still an ongoing program between NSF and NIH. And I know there's one, microbial observatories, which is basically, someone brought it up yesterday, microbial observatories is an example of a joint activity between USDA and NSF. So microbiome is on the lips of several agencies with actual concrete plans, although no timeline, concrete plans to have joint activities. And I think there may be real opportunities in those joint activities to address some of these environment/microbiome discussions.
Just a really quick clarifying question. There's a microbial observatory, which I haven't heard about. Is that at all linked to the National Ecosystem Observing Network that's being rolled out as we speak?
Well, unfortunately, for reasons that I won't go into, the Microbial Observatories program was sunsetted. It was an extremely successful program, and NSF decided to sunset it. It's kind of, unfortunately, indicative of that kind of period in NSF's history where they decided to kind of set aside almost all microbiological research.
Are you aware of NEON, the national?
Yes. And so
Maybe
NEON...
we can build something in there.
So NEON has now a microbial component to it, but more at sort of a monitoring level.
Yeah.
I don't know, because NEON isn't actually going to fund anything. It's just a superstructure onto which people can propose ideas. Yeah. Was there something else you wanted me to address?
Yeah, I mean, you started talking about a construct, concrete system, no timeline, no budget. But
right.
So how do we build? What would be some next steps for building some of these environmental intersections into that, and then to affect the next batch of non-existent monies that come in?
I don't know how to answer that, except I'm a rep on this 3-agency activity. I don't even know how one gets on there, but I think maybe somebody from NIEHS should be part of this working group. And maybe that's the one concrete way I can think of, is maybe someone like Lisa. Are you on that working group? Okay, well, there you go. That might be one way to do this. And we're in early days, so it's not like anything's set in stone. So perhaps that's how one concrete way we could do that. Can I make another comment, kind of unrelated in some ways?
I'll check with Mildred.
So I mentioned this yesterday, and I bet, I'm sorry I wasn't here yesterday afternoon, I bet Tom mentioned it. I mean, in fact, I think there's emerging knowledge coming from the microbiome that could help structure studies, and that is, that 3 enterotype concept, right? Because the enterotypes are built around not microbial composition, but the functions of the microbiome. And so functions meaning everything from, digesting food to processing xenobiotics. So one would think, and those are irrespective of, ethnicity, racial groups, countries, gender, age, anything. The enterotypes are a totally independent organizing principle of the microbiome. And because it's built around function and how the microbiome is functioning, it seems like that'd be an excellent way to structure studies.
Just to add to that, I agree. I think you could get results in 6 weeks from data that's already been collected, not yet published, simply by looking for the levels of mercury resistance genes across these samples. Right, it would be a data set that that would have immediacy and be a direct connection to the Human Microbiome Project and let you be an early mover. I mean, I would say that one of the most interesting things I've heard is that there's a signal that many years down the road that comes from whether somebody was or was not breastfed. And you can imagine the same signal persisting for an exposure to something like mercury, right? Interesting thing to look for.
I think this may be an appropriate time to say this, and I say this, half of my brain says this, the other half is screaming, no, don't say it. There could be some reason to focus research on humans. I think that when we move into, and maybe Vince would comment too, but I know from my experience in doing immunotoxicology, a lot of what we've done in mouse models is actually not correct and not transferable to humans. You know, we're talking about, although there's a great deal of evolutionary conservatism, there's also extraordinary divergence, both in the status of things related to the immune system as well as in its lability. And so, for example, most of the animal models of autoimmune disease have not been as informative as they should be. So I'm not certain that I'm opposed to a focus on humans in this way. I think some of the fundamental models of understanding change to perturbation, certainly one can study microbes in a variety of environments, even including petri dishes, to improve our knowledge. But I'm not so certain that we should necessarily bemoan a focus on humans if we're trying to figure out the human microbiome and how it might modulate risks of humans. We might want to know what's happening in important animal populations because they are important to us. But that would be not animals as models, but say chickens as chickens. So I just want to put that on the table, that maybe we could all understand that there are times when perhaps we don't understand the points of inference across some of these complex systems that allow us to exploit animal models more appropriately, but there may also be times, we've discovered this in our studies on mercury immunotoxicity, that the mouse models really were very misleading. And so we do all our work using human cells or humans.
That's actually very, and David, you might want to build on that.
This is David Balshaw, also from the Extramural Division at NIEHS. So Ellen's comments actually made me think, so we've done a fair amount of work with humanized mouse models where they have a, quote, human immune system. And I'm wondering how much ability we would have of taking nobiotic animal models and humanizing their microbiome as a tool to do some of the basic studies we could not do on humans in a, quote, semi-humanized system.
Well, I think Vince described some of those studies, but you're still putting it into, say, the mucosal immune system of the mouse. You may be transferring, actually, you know, the human microflora so that mice don't eat each other's feces, but maybe human feces. That really boggles the mind. But, you know, I still think we need to remember this is as Les said, you know, this is a mutualism with a cost system. We can't leave out the receiving environment when we're thinking about how the microbiome is interacting with some internal or external stressor. So that, I think, what I would say.
But I'm wondering, David, if some of this could easily, I mean, there were some, I think Vince brought it up, are there some additions, easy additions one could do to your, hopefully, eternally funded exposure
Hopefully
biology
eternally
program?
funded, yes. Let's pray.
Do you think there are opportunities one can?
Absolutely.
I mean, we already have the humans.
Yeah, so everyone should stay tuned to the council meeting next month and hear what we're thinking for the next round of the exposure biology program. But I think that, you know, it's not a topic we plan to include right now, but we certainly could include work focusing on the microbiota. We are talking about epigenetics in there. We certainly can talk about microbiota as well.
I think to not consider it would be actually a missed opportunity. And then
Indeed.
that and the national. So go ahead, John.
Ellen, I may channel the other side of your brain for a second. It seems to me maybe there are two different areas where to be thinking about here. One of which is understanding the interactions of the human microbiome and the environmental influences on it on human disease, where I agree, the animal models have been where I accept your expertise, the animal models have been misleading. But the other side of this that we've been talking about is the interpretation of the animal toxicology testing regime and being able to better understand the mixed results, as Vincent has alluded to. And the question is, right now we're very dependent on that for understanding, for reducing uncertainty in risk assessment, basically, of environmental chemicals. Question is, are we looking into the future? Are we moving away from that in such a way and moving into human-based cellular systems and acellular systems that is it not worth the investment to try to understand that by understanding the role of the animal microbiome on these tests that we're using so much for human risk assessment? Do we just kind of say, we didn't do such a great job, but let's just put that in the past and move on to a completely better system? Or do you think that in the next 20, 30 years, we're still going to be relying on that so much that we should understand and reduce the uncertainty?
I think that the answer, my own answer, would be somewhere in the middle, and I think Martin and other people might agree here that we should be moving towards the Tox 21 world. I mean, we have to, even for resource constraints, whatever, for a multitude of reasons. I think that animal toxicology has been, may continue to be very useful in kind of scoping the field. But if we really want to reduce uncertainty, I'm actually not interested in reducing uncertainty about Swiss Webster mice. And I think at that point, we have to think in a very inventive way about probably alternative in vitro high throughput systems, and we heard some remarkable things that are going on here that will help us decide how important will understanding the human microbiome be, and then using that to kind of bracket a range of uncertainty for humans.
And once we incorporate metabolism, we should be all set. Go ahead, George.
Thanks, Tina. So I raised my hand before John's question, but it was perfect segue, because I had raised my hand to really change the subject and talk about question 3, but I think I'll also be able to address the question on the table as well. And I guess, as we struggle to develop a consensus conceptual framework of what the microbiome is, we are talking a lot in metaphors. And one of the metaphors that I think is really helpful in addressing this question is the metaphor of the microbiome as another organ system. And if you think about it in that way, then this question of the absence of the microbiome in reductionist systems, in vitro systems, is no different than the absence of, say, a liver, or the absence of an endocrine system that provides distant signals. And these are things that are both limitations to in vitro systems, but also, if we know enough, opportunities to remove a lot of noise and just put the appropriate elements in, such that our experiments become more interpretable and not less interpretable. So that to me is, the fact that there is a microbiome and it does stuff doesn't negate the promise of using in vitro systems in a predictive system. The second thing is, as I think about where we're going in the future in toxicology, it's very much going to be a tiered approach that's going to build on 50 years worth of toxicology, most of which is in animals. And I think that that's actually an incredibly rich data source that actually, if we use it the right way, points us in the right direction as to which models, which reductionist models to use and how to use them and how to get interpretable data out of them. And the question is, how do we take that incredibly rich data source of 50 years worth of animal studies and parse it out such that it becomes more interpretable? We remove these not human relevant kinds of results and really focus on the others. And one of the things that I think has been brought up many times this morning is this idea of, are there, or is the microbiome from the traditional animal species and strains that we use for most of our toxicology testing. How does that relate to the human microbiome? And these studies like this have been going on back when we called it microflora for a long time, at least in the context of xenobiotic metabolism. But I think that it's only in the past couple of years that we've gotten to the point where we can do more than that and really from an omics basis look at the functional, the total functional capacity or functional capabilities of the microbiome. And it's really at that level that I think we can make reasonable comparisons, because if we try to do it organism by organism or enzyme by enzyme, we're going to get a lot of uninterpretable stuff. But if we can do it on a more global, functional level, I think that we can get some answers. And to me, and maybe I'm just jumping ahead to the data needs section, to me that's an area of potentially low-hanging fruit that could have a pretty big payout in terms of how we interpret the historical animal data set.
I think many of us apply this particular approach, is that when we have a problem, we use multiple lenses to look at it. And when you see the same picture through the multiple lenses, that's what you tend to focus on. I was talking with Les, we do that with our data as well. We slice and dice 16S and metagenomic data in multiple ways, all these named metrics of ecological this, that, or the other thing. And when the same message starts coming through, those are the messages we listen to first. And you can imagine that it's not exclusive of using animal data, be it humanized animals, conventional animals. You can imagine doing things in cell culture, as you pointed out. I know very little toxicology, as I pointed out, but I do remember them saying, for even in the Ames test or in human cell cultures, mixing things with liver microsomal preps for some period of time to let them stew and then seeing if it changes the activity in the cell culture. You can imagine doing the same thing with different, be it the enterotypes, whatever functional groups we have. We can say, okay, here's some of the main functions, and here are some sequence genomes of organisms that we know carry out these functions, and they'll form consortia with this other sequence organism. So this can be our activating mix. Or obviously that involves a lot of research, and that's the types of things that could potentially be funded when you're looking at what would be the next generation of toxicology testing, saying, well, what happens if we activate compounds or potentially detoxify compounds based on interaction with microbes. So I think that what we're getting at is this whole idea of not trying to come up with the perfect tool, but with a good suite of tools to carry out the functions that we're all very interested in and perform those functions.
That's what I was saying. I mean, even those simple things require resources. So if some of that thinking could be built into the funding structures, I think that's where it's going to make a difference. Go ahead.
Yeah, Ann Summers, University of Georgia. I want to speak to a couple of points that have been raised and connect them. One of them has to do with the public perception and the public enthusiasm for this, and I want to echo Lita's and Ellen's comments very, 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 very strongly. The public is out there very eager for knowledge on this particular area and waiting and with frustration at the degree to which the practicing medical community is ignorant of this and limited to diagnostic as well as therapeutic modalities that are unaware of it, to say the least. And I think that I realize that Dr. Cho didn't get a chance to include web information. I mean, that is the coin of communication now among the public. And they try to do a good job of educating themselves. There's some very, very good things out there that I think are microbiologically sound that I've looked at. There's some other just weird stuff where there's very, very clearly a need for education. But you are going to find an enormously receptive bunch of people out there. And somebody else mentioned the business about, I think it probably was, I don't know how they know about this, but anybody who has, I mean, most people have GI disturbances, and they are eager to find remedies, and they're out there doing their own experiments on the hoof. I mean, if you look at probiotic sales are going up, people really want answers to this kind of stuff, and I think that you will find them receptive. And from my experience in the area in which I work that has to do with the human impact of metal poisoning, consumer groups and patient groups, and if you look at the classical model at NIH of the Alzheimer's disease movement, okay, which was really founded as a way to get public input. And there's a book about this someplace that was founded as a way to get the public interested in this. Right now, I'm not suggesting that we go out and form public groups of pro-microbiome people, but the public wants to know about this, and the public are eager to influence the funding stream in respect of these kinds of conditions that affect them. So that's one point. And the other point is, well, who do you need to work on in terms of, and it's been alluded to, I don't want to step on anybody's toes, but I think that, I mean, I laud NIHS tremendously. Full disclosure, I think most modern disorders are a matter of intoxication of some kind, or at least are affected by intoxications of things that we seemed like a good idea at the time, and we've got too much BPA and other kinds of things in the environment that are affecting these things, including mercury. But I think that this experience comes from almost 10 years ago when I sat on a panel that reviewed a major program project thing to do, I think, twin studies in obesity. And there were a couple of microbiologists on the panel, there were endocrinologists, and there were gastroenterologists. And me and the other microbiologists got it. And the people who were taking exception to the idea that this could have any explanatory power whatsoever were the endocrinologists and the gastroenterologists. And I think, I don't know that that's changed. I'm delighted that NIHS is taking the lead on this, but I think in terms of where we go in terms of shifting the funding stream, we need to have people at tables like this from MIDDK, from NINDS, and from the other fiefdoms in health research who are aware of this, not just at NSF and USDA, I think, because it's one health, and that sort of, I want to connect those two things. So, yeah.
Steve, I was going to pick on you, and then Marty. Lauren, do you want to also say something? Okay, we'll
yeah, this is Steve Rappaport from Berkeley.
I missed you.
In research needs related to the microbiome, what we've heard are a lot of interesting ideas for doing advancing experimental work to understand the diversity and function of the microbiome. Another strategy that I think could have more immediate payoffs would be to focus on biomonitoring in human populations and exploiting the current technologies in metabolomics. I was struck yesterday by the generation of small molecules by the microbiome, and these small molecules are inserted into the circulation of the host. They can be monitored, and there are several very recent examples, including the one I mentioned yesterday of Wang and Hansen's paper in Nature, where metabolomics basically discovered risk factors for cardiovascular disease that were followed up with experimental studies in animals. There's very similar work that's been done by Nicholson and Elaine Holmes in London. There are not a lot of investigators that do this kind of work, and I think by encouraging it, we would learn a tremendous amount about the microbiome and about other sources of environmental exposure, including diet. So I would just like to make a plug for that.
Perfect. Marty, go ahead.
I just wanted to return briefly to the points that Ellen and George were making about how to apply this microbiome research to future research, whether it be in vitro or in animals. And I have some sympathy with what George is suggesting, that in addition to thinking about the intersection of the microbiome and question 3 there, the toxicity pathway approach, that we also give some thought to how applying microbiome type thinking can help us better interpret the animal studies from the last 50 years, particularly at the 10,000 foot level. But I would hate to see that become a major research program. It just, I don't know how much we will be able to use those results to interpret results from laboratories 30, 40, 50 years ago. If you look at the president's budget for EPA, I mean, they're putting extra money into computational approaches for pathway sort of testing, toxicity testing. And that seems to be the future. That's the future as envisioned in the National Academy of Sciences report from 2007. We can't have a complete break with the past. I understand that, but I don't know that there'd be a lot of enthusiasm for major investments into microflora studies on how to better interpret the results of animal studies from decades ago. So if I were controlling the purse strings, I'd certainly put much more money into the pathway approaches. I could see how the animal studies are going to continue to be a significant part in basic biomedical research, and perhaps there one needs better interpretation of the impact of microflora on the results.
Perfect. Lauren, Richard, and Les.
I guess building on this last point, I think even though a lot might not be gained by having sort of an in-depth approach trying to sort out the differences in microflora and the impact on an ongoing experimental basis, there's probably a lot that can be gained by data mining. We have this whole history of many different agents tested in a variety of different animal organisms. Just thinking about some of the risk assessment work we perform, there's an incredible amount of quantitative consistency across a variety of animal strains. And I think it probably will be the case that for certain molecules, certain types of molecules, the microbiome will be very, very important. Metals comes to mind. There's a lot of inconsistency when we think of human and animal in overall results in a risk assessment sense for metals. But for some molecules, oral route, inhalation route, if you do things on a dose basis, you go across animal strains and species, there's a lot of consistency. And I think it would be useful to try to get a handle on the extent of that. And again, it doesn't require new experimentation. It requires some data mining and comparisons. And of course, then the FDA has an incredible amount of animal and human data that could be looked at. I guess the other thing that I'm having trouble seeing the way forward, and it's really a question to some of the researchers, inter-individual human variability is an incredibly important issue for public health control of environmental agents. And I'm not seeing an approach to kind of address that in the context of interpreting pathway testing. And I'm just sort of blanking on what kind of strategies might be possible to begin to address this issue of inter-individual variability. It's sort of the susceptible individuals that are driving the environmental risk. So it's really a question to the researchers.
I'll let you directly address this.
I guess just a comment on that point. You know, we are planning a workshop on inter-individual variability. We're sort of in the middle of crafting an agenda for that meeting right now. It will be next February or so.
Right.
So, you know, I think that if there is a possibility of following up with some of the people around the table and talking about how we might begin to address some of the issues as a follow-on to this discussion on the microbiome, it would be very helpful for us.
Richard, go ahead.
Richard Denison, Environmental Fund. Lauren just raised one point I was thinking about too in terms of the variability issue, and I think thinking about the microbiome with regard to both being a source of variability that may explain, for example, a lot of the different conflicting results in studies, but also an additional contributor to variability that needs to be accounted for in determining whether, for example, when you look across a whole human population, concepts like thresholds, safety thresholds and so forth, apply, and that's been a controversial topic that the Academy has addressed in a prior report that Lauren was intimately involved with. I also wanted to touch back on the issue of consumer perception and the drivers here, or public perception and the drivers. I had an opportunity a couple of years ago to meet with a number of consumer product companies and to talk with their scientists about what was driving them to market this broad array of personal care and household products that contain antimicrobials. And to a company, the scientists in those companies sort of hung their heads and said, marketing made us do it, essentially. That the efficacy of those, the consumer benefit, if you will, was simply not apparent to those scientists in those companies, and yet they were marketing these broadly because of the public perception that had been created. So it kind of makes me think of the way the environmental community had to do a huge rehabilitation of wetlands about 20 years ago to stop people from thinking about wetlands as swamps that were mired in disease pits and snakes and alligators, and turn them into a more positive view of what they are and how they function, services they provide, et cetera. I think we need a similar rehabilitation of the microbiome, perhaps, in humans, to get people to thinking about it in ways that are less simplistic. So that's a countervailing view to what I agree with Anne having said earlier, that there are some other drivers that are providing a positive view of the microbiome. But there's this negative, I think, view that we have to protect ourselves from our own flora that is driving a lot of perception as well.
They're probably both true. Les, and then Ellen, and then, Trey, sorry, and then Trey.
Yeah, I have a couple of thoughts to share. One of which, coming back to this questions about interhuman variability. And Lita, I apologize. I want to highlight something that you've mentioned about this recent study by Rumagam that just came out in Nature. If I'm correct in my reading of that, and people ought to read it for themselves, I think his analysis is based on metagenomic reads, so they're the full genome. But the classification of the three enterotypes is not primarily functional. The classification is based on a taxonomic identification of those reads. And while the taxonomic separation of these three enterotypes is quite clear, then they went to say, are there functional differences? And there are, but they're fairly small and subtle. So in a way, it mimics my own finding that a perturbation effect that's quite obvious in the phylogenetic analysis of the reads, it's not absent, but it's much smaller in magnitude looking at the functions. So it could be that there certainly are functional differences among these three enterotypes, but that wasn't the primary way that those enterotypes were discovered or defined. There's a suggestion in that paper as well that would provide a very strong functional basis, and it could be that there's three different pathways for ultimate fate of hydrogen among methanogens, methanogenesis, sulfate reduction, or acetogenesis, which, if that is in fact the basis of the three enterotypes, it would be sort of this beautiful, elegant explanation. But that's not yet clear. And also, with respect to that, I would encourage people to read an accompanying, well, it's not an accompanying paper, but a paper from De Filippo, published in PNAS in 2010, where they compared the Italian children to children from Burkina Faso. And I would love to see where these Burkina Faso children find themselves in those three enterotypes, or in fact, if there may not be a fourth. And as we continue to study people outside the developed world, whether there will be a longer and longer list. But if you're interested in the questions about interhuman variability and questions about the internationalization of testing, that PNAS paper in 2010 from De Filippo is, I would say, a must-read. With respect to public perceptions of all this, I've had a recent introduction to this. I'm not a physician. I don't deal with patients. I think of myself kind of as this scientist following my own interest stuff. But after I had a paper published last fall that generated members of the public getting a hold of me with their interests and concerns. And I found this very interesting. There certainly is a subset of the population that's entirely dialed in, and they're getting it, And they are people, as you mentioned, Sue, that have problems. People with ulcerative colitis. My paper, I didn't study diseased people. I didn't have anything to do with inflammatory bowel disease research. And people are getting a hold of me saying, oh my God, you mean if I took an antibiotic, I may have made a permanent change in my microbiota? And these are people who have been living with UC for years and who suspect that, in fact, some perturbation like that was, in fact, their origin. And these people are totally driven in their life. I suppose if I had, a couple dozen bowel movements a day, it might affect my life too. These people are driven to manage their microbiota. And they know that's what they're doing, and they're frustrated that they don't have any more tools to do it with. But when they are on these fairly restrictive diets with the SCD, specific carbohydrate diet, they have this sense that I am trying to feed the bugs that are helping me out and starve the bugs that are making me sick. And that may be exactly the nature of their disease and ultimately the nature of a successful treatment. We don't know that yet. But I had conversations with members of the public that were highly motivated and reasonably aware of the science and who have blogs that they put up links to my paper and many other papers because of that. And there's sort of an intersection of this group in the public, along with perhaps the generation of people who grew up after Silent Spring, who are prepared to look at humans as an environment that is in need of care and remediation, and to some extent, as it may often be the case, there's a little bit of irrational anti-science, kind of anti-technology bent towards, well, there must be some kind of natural Eden which we can get back to, and if only I bought the right probiotic, I'd find my inner Eden. So there is perhaps some science and education that we need to do there also to encourage or tap into this sense that yes, we need to manage human health more like managing an ecosystem, but also acknowledge that that doesn't mean that if only we let it go natural, everything will be okay, and there'll be no more, cancer or old age or anything like that. But I think this is an arena where there's veins of public interest and motivation that are they're present, they're active, and if we can tap into that, we'll be doing both ourselves and the public a great deal of service.
Ellen, did you want to directly respond?
I wanted to talk about the variability issue and just make a suggestion to Bill and others. There's a really elegant paper that was written by Leslie Stainer and Paul Schulte from NIOSH, which essentially tried to answer the question of how big a difference would make a difference in terms of policy. And they provided a very nice metric. It was really in the context of gene-environment interactions. Say, okay, you have now determined some polymorphism, some SNP or whatever, that seems to be relatively prevalent in the population. Functionally, how big a difference does it make? And they proposed, and in the scale of how we do risk assessment, which builds in all kinds of uncertainty factors and protective and conservative assumptions. And what they really pointed out, I think in a way that maybe as Lisa said, we're all realizing to our chagrin that most of these changes that are encoded genetically probably don't add to a whole lot in terms of the variability. And so I would advise, I think it might be useful reading for the committee because it's sort of a roadmap in how to think in a meta-scientific way about all these fascinating sources of potential variability. But that's why I reacted so strongly to Tom's paper, because those differences in the reduction, oxidation, methylation, those are enormous differences. They're much bigger than the differences encoded by silly little polymorphisms in GST omega in the arsenic metabolic pathway. I mean, those made me, really sit up and take notice. But other than just having a personal reaction, I think Leslie's paper is very, very useful.
Trey, go ahead, and then Vince, and we might have to wrap it up at that point.
Okay, I just had a very general observation. I think it goes to question one. Consumer Product Safety Commission, we deal directly with the public, and many respects. And I think just the technology and the way that information can be disseminated, and unfortunately sometimes even misinformation. So I think in terms of the public perception, you know, we have YouTube. It was really almost surreal. One of our risk assessments, there was a guy on YouTube talking about it and going through it. You know, there is also another person from a website that, again, the same risk assessment, walked through it and did a critique of it, and actually it was pretty good. But, you know, with Twitter, with, you know, the iPhone, that type of technology, I mean, information and concerns about chemical exposures and just diseases in general, I think can really go to the public much more quickly. So I think that, again, not just specifically to the microbiome, but just in general, it's just amazing over the past 10, even 5 years, the ability to get information out to the public is really critical. And I think that the public also will react, write letters to their representatives. So I think that too can play a role in science policy in the way that we do. The questions will be asked. So I think that, you know, again, it's not just and that's where I think having the correct information that they're then passing on to local, state, and certainly federal representatives is critical. And the questions that they then ask us, I think certainly as regulatory and even perhaps even research agencies in terms of the direction that we're going.
You may have the last word.
So with regards to the media and public perception, you know, as a physician, I've had some interesting interactions about microbiome research. And I was fortunate as an assistant professor to have several days, basically a week of media training as an assistant professor when I was at Michigan State University, and it served me well. But then I realized how well I didn't learn some of the messages. For example, one of the things that I study Clostridium difficile recurrence as a problem, and to sort of show the futility and how little we know about the microbiome, I will often make a comment about how fecal transplant is useful and is widely used in their randomized controlled trials. And you figure out how you randomize stool and control it. But when I mention these studies, and I make sure, and I go about 5 times, I talk about as showing sort of the scientific ignorance, the fact that we resort to that now and things like that. I will always, and I stopped doing this a little because I always, for the next few months, I will get lots of calls and emails and letters, impassioned emails to say, Dr. Young, I want to enroll in your stool transfer therapy. You know, even though I stated it up and down, and when I proofread the articles, I make sure that it's very clear that I'm not encouraging this and very trying to make sure that I don't do it. It doesn't matter, you know. So the public is really, these people who are suffering from these diseases are really, you know, that it doesn't seem unusual to them. You know, fecal transplant makes perfect sense, right? So that's what we have to, you know, deal with, and I think it'd be great if we could come up with something short of that. And the final comment with regard to that, someone made the comment that we've kind of rehabilitated the idea of conservation, and I wrote a review with a real live ecologist. So we had an ecologist and a biomedical researcher writing about the microbiome and how to bring in both fields of research. And our final section was talking about war no more, you know, that we're so put up on the pathogen and this destruction of pathogen in that analogy, and that maybe the idea of national parks and conservation with regards to the microbiome is how we need to go. And we've both gotten a lot of very positive feedback. And it's interesting, I get feedback from the people who are biomedical researchers that they understand the ecology side. And then the ecologist has told me when we met at a meeting that the ecologists are going to, now I understand why these biomedical researchers are interested in this and how we can help them. So I think trying to get the message out there is important. We're trying to do it in a number of different ways, and I think we can't stop doing that.
Thank you. I know I said you have the last word. Having heard all the discussion so far, is there another parameter, another point you want to add to the discussion, especially with the topic that you spoke about this morning?
Yeah, I do really agree with those comments and this idea about sort of trying to come up with another metaphor to rehabilitate this notion, because I do agree I think there's a lot of interest in the general public and that people do get this idea. I mean, everybody's seen the Dannon yogurt commercials and they kind of get the idea of there's good bugs and bad bugs. And the little old lady who's the little Russian lady, you remember that commercial? That's from like the 70s. People get that notion and they're really interested in it. So I think that thinking about not just risks, but benefits. Microbiome researchers, I think, have a much easier time of convincing people that they're working toward a benefit that everyone would agree is a benefit. They may not know how to do it, but I think everyone's kind of on the same page, as opposed to, say, nanotechnologists, or, I think there's less fear of the microbiome community of researchers, but I do think it's important to think about how being very clear about sort of the benefits issue, and also in terms of this sort of rehabilitation notion. I think it's very difficult. It'll be very difficult to do, but very important to try to come up with the equivalent of wetlands for swamps. Because I know I've talked with researchers who are investigating the hygiene hypothesis, for example, and they're very into this idea that dirt might be good. And yet, when I ask them, do you use triclosan soap, they all say yes. And so I don't know where that comes from, but there's this weird, even in the scientific community, this kind of dichotomy of practice and sort of beliefs at a certain level that needs to be addressed.
So with that, I want to thank Mildred and the morning panelists and everyone who contributed to an excellent discussion. And Ellen, top that.